Hi, I'm Dr. Ben Chapman. I'm the director of the Food COVID Net project. And thank you for joining us today for our second webinar in a series of webinars that we'll be putting on over the next couple of years that are focused on COVID-19 challenges in the food sector. And today's webinar is one that we, as a project, many of our stakeholders have reached out to us to talk about this and to hopefully develop some engagement around and fill some data gaps in. But it's really around airflow, ventilation, and close contact in the food sector. We're really excited today to have two just phenomenal speakers to share their expertise and hopefully answer many of the questions that are out there. Before I pass things over to them, I just want to give a very quick background about what Food COVID Net is for those of you, if this is your first webinar joining us, first time coming across this information. We're a project that is a collaboration of researchers and extension and outreach individuals in the food sector at NC State University, the University of Florida, Rutgers University, and the University of Nebraska at Lincoln. The goal of our project is really to fill data gaps and information gaps around COVID-19 management, as well as understanding transmission, persistence, inactivation of SARS-CoV-2, specifically in the food sector. So our project started in October of 2020, and over the next couple of years, we'll be conducting research to fill data gaps and also conducting much more outreach like what you're participating in today. We're, as I mentioned before, we're really excited for today's panelists and for presentations today. It's an area from all the way from packing facilities through to restaurants that we've seen over the past year a whole bunch of questions about. And what's really exciting for us is to be able to get two researchers that are working explicitly in this area and bring them into the food world today for the next hour. Just a little bit of housekeeping, how this will work. Dr. Angela Rasmussen is going to give us a little bit of an introduction to the topic. Tehya Stockman is going to also talk about airflow. Angie's going to talk about close contact and what we know, sort of the cutting edge science in this area and where it fits into food. If you have questions, and we received many questions through registration, so if you have questions, we do have a question and answer function. Please continue to populate that with any questions that you may have, and we'll run through as many of these as possible at the end of our session in a panel Q&A. With that, I'd like to pass things over to Dr. Angela Rasmussen. Dr. Rasmussen's a virologist studying host responses to infection by combining classical virology with modern systems biology approaches. Her research objectives are to identify host response signatures, predictive of infection severity or disease outcome, and host pathways to target drug development or repurposing. Dr. Rasmussen has published numerous original articles in the peer-reviewed literature, serves on the editorial board of Cell Reports and MSphere. In addition to her scientific work, and really this is where I've learned about much of what she does, she believes that engagement of the public is essential to successful public health initiatives, and is extremely active and outspoken science communicator. So really, that's one of the biggest reasons why we invited her today. Is just she's a real leader in this area, and especially talking about it with diverse populations. And so, Angie, please take it away. The floor is yours.
Thank you so much for that kind introduction, Dr. Chapman. And I'll just jump right into sharing my screen. Sorry about this. You'd think that I'd be faster on the draw with Zoom these days. Okay. Is this the full screen view that you can see?
We've got the presenters.
Okay,
But
got it.
yeah.
I will swap.
I had the exact same problem. I don't know if it's a new like a Zoom situation, Angie, but when I started, we also ran into the same issue.
Yeah, I have
There we
two
go.
desktops,
Yep.
and I never know which one is actually being shared on the Zoom screen when I do this. So I've actually given entire presentations in the presenter mode by accident without realizing it. So I'm just going to get started. As Dr. Chapman said, I'm Angie Rasmussen. I'm currently affiliated with the Georgetown Center for Global Health Science and Security, and I will retain that affiliation. But early next month, I'm moving to Canada and starting my own lab at Vito Intervac, which is a vaccine research institute at the University of Saskatchewan that's building a really exciting emerging viruses research center there. So let me just jump right into this. Before I get started talking about airflow and the virology really of transmission of COVID-19, I like to start with the territorial acknowledgement and equity statement. And today I'm in Seattle presenting from the unceded ancestral homelands of the Duwamish people. I acknowledge and honor the first people of these territories and their tribal governments, their histories and ancestry, and their role today in caring for these lands. And I'd also like to acknowledge the history of systemic inequity in academic science that spans centuries. My prior institution, where I was faculty and where I got my PhD, Columbia University, and my current institution, Georgetown University, were both founded using profits from the transatlantic slave trade and the sale of enslaved people. And in addition, they excluded women and people of color from the academic community for more than 200 years. And this has left a long and painful legacy of racial and gender-based inequality that continues to this day, as well as throughout academia. I encourage all of the folks listening today to consider how you can contribute to making scientific research and biomedical research a more equitable enterprise. So to get started, as most of us know, SARS coronavirus 2 is a respiratory virus. It does infect other organ systems in the body, but where it primarily infects and causes disease, or classical COVID-19, is in the respiratory tract. So the nose, the sinuses, the trachea, the oropharynx, and the lungs. And on the left here, this is a CT image from a COVID patient showing the difference between normal lungs on the left and severe COVID-19 pneumonia on the right. You can see these very characteristic white ground glass opacities that are visible on this radiographic scan. On the right, I have shown some images of lungs from the rhesus macaque model, which is a model of moderate COVID-19 that I work with. And you can see in this model, you can detect on chest x-rays areas of infiltration of immune cells that's indicative of pneumonia, as well as the gross pathology on the bottom there. Sorry for the graphic image, but these are what the lungs look like after the animal is necropsied on day 3. And you can see these large visible lesions on the surface of the lung. So SARS coronavirus 2, again, does infect other organ systems, but it primarily infects and causes disease within the respiratory tract. So many cases of COVID-19, the majority of cases of COVID-19 have impact on the respiratory system. Now, respiratory viruses are transmitted by different routes of transmission, different modes of transmission. For SARS coronavirus 2, the most common is inhalation of aerosols or small respiratory particles that are in the air, as well as direct contact, or what's sometimes called droplet transmission. These are larger respiratory particles that can be expelled onto somebody's hands. Sometimes they can be expelled onto a surface as well. That's a less common route of transmission, but direct contact transmission very much likely happens in addition to transmission by inhalation of smaller particles. Less common are routes that we know can be efficient routes of transmission experimentally, but we don't have as much evidence about them happening in the real world. And the epidemiology evidence suggests that they probably aren't as common as inhalation and contact transmission. And that's ocular transmission. So through the eyes, we know that the eyes express ACE2, the receptor for the virus. And also animals have been effectively infected in experiments by ocular exposure. We know that oral exposure can sometimes contribute to this because the mouth is connected to the oropharynx, also goes down into the respiratory tract. Although it's really important to note here that since this is a food industry seminar or food science seminar, that this virus cannot be transmitted, as far as we know, by ingestion. So orally, it can find another way to the respiratory tract, but if you actually eat SARS coronavirus 2, it's unlikely to cause infection that way because of the low pH environment in the stomach. Likely degrades most of the infectious virus particles. Now, while there has been evidence of virus, including infectious virus in feces, this is primarily a risk for either contact transmission or inhalation of bioaerosols. And that has not been really proven in any circumstances. There are a couple case reports that link possible inhalation of fecal bioaerosols to defective plumbing and defective water lines, but it's not thought that the fecal route is a major source of transmission either. And then finally, as I mentioned before, indirect contact or fomites. This is when a droplet or an aerosol will settle on a surface, and then you touch that surface and then touch your nose. And this has also been experimentally demonstrated in animal studies, but it actually is less efficient, much less efficient than experimental aerosol transmission or experimental droplet transmission. So we think that fomites are also happening less frequently in the real world. And it's notable to say that this is actually different from some other respiratory viruses. So rhinovirus, which I studied back in graduate school, is transmitted much more efficiently via fomites. And that may actually have to do with the fact that it's a different virus. It's a non-enveloped virus, so it's increasingly stable in the environment for a longer period of time than SARS coronavirus 2. But again, fomites possible. We should still take mitigation measures to prevent fomite transmission, but they're not the main route of transmission. So another key concept that I kind of wanted to explain to everybody is the concept of susceptibility and permissivity. A lot of people think that if you are exposed to a single infectious virus particle, that's automatically going to infect you. And the reality is a virus has to be in a very specific situation in order to cause an infection. And it has to meet up with a cell that's both susceptible and permissive to infection. So when we talk about susceptibility in the virology world, we're really talking about cell entry. And that means that the virus has to come into contact with a cell that's expressing, first and foremost, the cellular receptor, as well as any necessary cofactors for that virus. In the case of SARS coronavirus 2, the receptor is human ACE2. There are also cofactors that are required for what's called priming. This is processing of the spike protein prior to it being internalized in an endosomal compartment. And that is done by a molecule called TMPRSS2, which is a protease that's on the surface of the cell. Now, it's possible there may be other cofactors that can substitute for TMPRSS2. You can induce this experimentally by treating cells with trypsin, for example. But TMPRSS2 is anyways the one that we know about, and that is expressed on the surface of certain cells in the respiratory tract, and specifically type 2 pneumocytes or type 2 alveolar cells. So a virus has to get in touch with a cell that's susceptible. But what happens after it comes into the cell? Well, there's as this diagram shows, there's all these different steps that then happen in terms of the virus replication cycle. The virus undergoes what's called fusion, in which it expels its genome from the endosomal compartment that it's in into the cytoplasm, where it then starts to be transcribed. It also begins to be translated, or at least the ORF1ab polyprotein begins to be translated. The RNA-dependent RNA polymerase, or RDRP, begins making the subgenomic RNAs. Those begin to be translated, and then all of those viral proteins work together to assemble new progeny virions, and those exit the cell. all of these processes, as many people know, require the host's involvement. When people talk about how a virus hijacks a host cell, this is what they're referring to. So the virus has to rely on cellular proteins or other cellular components to carry out its entire replication cycle. And sometimes in certain cell types, that means that a required host cofactor needed for replication, assembly, or egress won't be expressed. And in that case, even if the virus can get into the cell, that cell is not going to be what we call permissive to infection. So it's not going to result in progeny infectious virions budding out of the cell. It's going to be an abortive replication cycle that hits a dead end at some point. So that's one barrier that viruses have to get through in order to establish an infection. And it's really important to think about that with regards to transmission, because that really determines which route is going to be most likely for a virus to infect a new host. Now, there are a number of other barriers to transmission. And one of the ways that virologists tend to think about this is through minimum infectious dose. So this slide really simplifies a very complex topic, but the minimum infectious dose is basically the minimum number of infectious virions that you need to be exposed to in order for a productive infection to be established. Now, in theory, a lot of people think that, oh, one infectious virus particle should be enough to start an infection, and that's true. And certainly that's true in cell culture, where a lot of these barriers have been removed. But in the real world, it's more complicated. And one of the reasons why it's more complicated is that RNA viruses especially often make a lot of mistakes when they're replicating. So you will have a big pool of virus particles, but not all of them will be infectious. They might be what we call defective interfering particles. This is a particle that either doesn't have a virus genome in it or has a defective genome in it. And these can compete for receptor binding, for example. They can also stimulate the innate antiviral immune system. And they can be present sometimes in great numbers compared to infectious virus particles. So this cartoon that I made, because I didn't want to cut and paste a million virus particles onto this PowerPoint slide, I've just shown a particle to PFU ratio of 2. That's the number of non-infectious particles to infectious particles, or plaque forming units, or PFU. That's how virologists measure infectious virus. So this is showing a particle to PFU ratio of 2. There's 2 non-infectious particles for every infectious particle in this exposure dose. Now, the first barrier that's going to hit is going to be the environment. So is it in an environment where these infectious particles can stay infectious for a longer period of time? Is it in a situation where there's other people around? If a virus is, expelled onto a surface or into the air in a place where there's no new hosts or no susceptible hosts, then it's not going to be able to cause an infection. So that's the first barrier that it's going to hit. Oops. You're going to lose some of these particles along the way as you go through each of these series of barriers. The next barrier it's going to hit, so let's say this dose of virus is in a place where there is another person working, and that person is susceptible, and it gets into their nose through either direct contact or through inhaling aerosols. Then it's going to hit physical barriers, and that's your nose hairs, the mucus that's produced. That the purpose of that is to provide a barrier that pathogens can't get across. So you're going to lose some of the non-infectious and infectious particles when it meets that barrier as well. Then it has to come into contact with a susceptible and permissive cell. Now, if you look in cell culture, you see beautiful cytopathic effect when you just put a very small amount of virus onto a monolayer of vero cells, for example. But that's an entire plate of cells in which they're all susceptible and permissive to infection. In the nose, it's a 3-dimensional architecture that's composed of a variety of different types of cells, all of which will be producing different factors that may or may not be required for viral infection. So you're going to also only have a smaller percentage of the viruses in your initial dose actually come into contact with the cell that's both susceptible to infection and permissive to replication. And then the final barrier that it's going to hit is the innate antiviral immune system. So we've evolved a number of different sensors, if you like, to detect RNA virus infection. They detect motifs that are associated with RNA virus infection, but not with normal host cellular processes, such as double-stranded RNA. CpG is another one. There's a number of different cytoplasmic and endosomal receptors that can detect virus genomes or the products of viral replication, and this will trigger the interferon system. The interferon system then will go on to trigger what are called interferon stimulated genes. And these ISGs, or interferon stimulated genes, will sometimes directly antagonize different parts of the viral replication process. So they can insert their own blocks effectively to the replication cycle. After all of that, if you still have an infectious virus particle around, yes, then that will establish an infection. That virus will replicate productively, and you will see intrahost spread to other susceptible cells. And eventually, you will start to see viral shedding, and then you will see the progeny viruses go out in the respiratory particles that are emitted by that host, and hopefully for the virus, infect another person, not hopefully for us. So when we're thinking about mitigation measures in any environment, but in the food industry or in settings of food preparation or food processing, we need to be thinking about mitigation measures that will add more barriers up front before you hit all of these biological barriers that can reduce exposure dose. So if your minimum infectious dose is, let's say, 100 infectious virus particles, if you add ventilation, if you add masks and things like that up front, then you're basically you're raising the minimum infectious dose. You make it so that there are more virus particles that have to be present in order to overcome those other barriers that you're adding in terms of mitigation efforts. And eventually, with any virus, viruses cannot sort of produce unlimited amounts of virus. You will get to the point where you've just made the minimum infectious dose so high that it's going to be impossible for the virus to establish an infection. And that's why when we think about mitigation measures, we really do need to think about how additive they are. So masks alone are great. Masks with ventilation are even better, with physical distancing in certain environments, with good hand hygiene. All of the above are going to increase the number of barriers up front that the virus has to overcome in order to infect a new host and be effectively transmitted. So I'm just going to end with this slide that says something that everybody here probably already knows, that many high-risk situations exist in the food industry. Now, one thing about SARS coronavirus 2 that has become apparent from an epidemiological perspective is that transmission is very overdispersed, which means that not everybody who gets infected with SARS coronavirus 2 is necessarily going to transmit to another person. This means that transmission is very situational. And so what we want to do is try to mitigate these high risk situations, and by raising that minimum infectious dose requirement, as I talked about in the previous slide. So in the food industry, for indoor dining, there are no masks, there's prolonged close contact, there's variable ventilation, increased salivation when people are eating, and there may be food sharing. And all of these things can contribute to transmission risk. In kitchens, masks can be difficult to wear. It can be an uncomfortable environment for people to wear masks in. People are in prolonged close contact. Ventilation is very variable, and there are many high-touch surfaces that people might be exposed to. And then finally, in food processing plants, we've seen many outbreaks in meat packing plants in particular, and that's because, again, they are a high-risk situation because of their very nature. There's prolonged close contact on the line. There's variable ventilation in these facilities. There may not be masks. There's cold temperatures, which experimentally has been shown to preserve infectivity over a longer period of time. And there's difficulty maintaining hand hygiene over time because people are obviously handling all of this meat, and they're not getting breaks to go wash their hands and step away from their colleagues. So when we're thinking about how airflow and ventilation could improve this, we really should be thinking about incorporating as many mitigation measures as possible along with that. Because these situations are very, very high risk, and they are very conducive to transmission of this particular virus, SARS coronavirus 2. And with that, I'll stop. And I really look forward to hearing Tehya talk about ventilation and airflow, because that is not my area of expertise. So I really looking forward to learning from her.
Great. Angie, thanks so much. And just as a reminder for our audience members today, as you have questions, please put them into the Q&A, and we'll have lots of time to talk about that. So our next presenter is Tehya Stockman. Tehya's a PhD student at the University of Colorado Boulder, studying environmental engineering with a focus on air quality. Since June 2020, she's been working with musicians to measure aerosol emissions for musical instruments to understand COVID-19 risk and how to mitigate risk. And so Tehya, the floor is yours.
Awesome, thank you for the introduction. And I will share my screen. Can you all see that?
We absolutely can.
Awesome. So today I'm going to be talking about the more control measure side of things. So how can we control an airborne transmitted disease? Let's see. So first of all, I wanted to just make sure that everyone has an idea of what aerosols actually are. So because there's been a whole lot of misunderstandings about what actually aerosols are, and they are just simply solids and liquids that are suspended in a gas. So anything from like what you have out of an aerosol can, to smog, to smoke, viruses that are suspended in the air, bacteria in the air, and even pollen, which we know can be really, really large and can be suspended in the air outside for very long distances, because that's how plants actually reproduce. So you can see that the size range of aerosols is really, really large. So this is 7 orders of magnitude. So really the control methods that we have is trying to think about how these different orders of magnitude can be controlled with these different devices. So aerosols, as I said, can stay in the air for a really long time. Like if you've ever had a pollen warning, if you suffer from allergies, that's because those pollen is an aerosol that you inhale outside. That is like a large concentration outside. Aerosols can also become suspended in a room, which is what we're really focusing on today, because for COVID, a lot of the transmission has been indoors, and this is because those aerosols, what happens is the room becomes well mixed, and this one source of someone talking can then distribute across the room. So while these large, more like spit-like drops fall to the ground, aerosols really can stay suspended in the air much longer and can even be resuspended in the air from movement in the room. There's been a lot of evidence for airborne transmission of SARS-CoV-2. So I just wanted to show, for example, there have been studies in hospitals where they're able to sample the air in different locations around patients' rooms and have found air samples containing SARS-CoV-2. And then one of the largest ones, which led my team to focus on musicians for our work, was the Skagit Valley Choir that happened back last March. Over a 2.5-hour rehearsal, 53 out of 61 choir members contracted COVID, even though they were not all directly next to each other. There was definitely some space in the room, which suggested that these aerosols were able to build up in the room enough for other people to get infected. So why is social distancing even important if aerosols are the concern? Well, what I've seen a lot in my research studying musicians is very similar to this image here, where you have a person sneezing and you can see how those particles disperse with distance. So when you're in a room, the reason why we're saying that, 2 meters or 6 feet from each other is because you have a big cloud of initial aerosol that you're producing, and then those rapidly mix in the room until what we consider is in the aerosol world, a completely mixed room situation. So these aerosols are much more homogeneously mixed after these so you have the close proximity, but then these aerosols then disperse relatively evenly throughout the space, which is why we need to think about the entire room when we're looking at control methods. So one thing that I wanted to also touch on is, how do masks actually get at these aerosols that are extremely small? Like, how does it, these respiratory aerosols containing the virus are are larger than SARS-CoV-2, of course, because they need to contain it, but they're much smaller than a lot of these things. And I've seen a lot of people thinking that a mask works similarly to like a colander, because that just makes intuitive sense. Like, the big stuff is filtered out and the small stuff still gets through, but that's not actually how it works. So how a mask works is that masks are full of fibers. You have airflow that goes around these fibers. And what happens is these small particles don't really care exactly where that airflow is happening. There's also this random motion that happens, and eventually they'll just stick to a fiber. And the mid-sized particles follow this streamline, but then they might brush up to the particle or might not, which is why sometimes these more intermittent sized particles are actually more difficult to control. And the large particles, they're just like, I don't care about the streamline. They have a lot of mass and a lot of inertia, and they just ram into these fibers. So they are easy to filter out. So it's really these mid-sized particles that are the most important for long-term air. They end up staying suspended in the air the longest, and they're also the hardest to control, which is why, people have been talking about N95 masks. Well, they're good at controlling these, especially these mid-sized particles, but then also the small and then large ones as well. It's important to note that masks filter aerosols, but they also stop these large projected jets, like what I showed on the previous slide, from actually just hitting someone else's face. So when I've been doing a lot of these musical instrument testing, I've seen that these jets are greatly diminished, which means that you have much more rapid mixing around someone's face, and you're less likely to have a lot of aerosols from someone else going into your face, because that air is becoming much more mixed with the surroundings. What's also important, though, is mask fit. So sure, an ill-fitting mask is still better than no mask, because you're still stopping a lot of that airflow and a lot of those particles. However, aerosols, they like going around things, and they follow the streamlines very easily. So you're going to have aerosols leaking out of places in the mask where it's not as well fit. So some masks are better than others. So for instance, this pink is a surgical mask, and this is tested by Lindsay Marr's group. You can see different types of masks are more effective than others, but then also, for instance, a bandana with 2-ply is better than a bandana with 1-ply. But this is not taking into account also then mask fit, which is also really important. And if you have a mask that's really hard to breathe through, and you have these like gaps along the side of your face, the air is more likely to go through those gaps, and the mask will not actually be as effective. So as I said before, aerosols can build up in a room over time, which is why it's important for proper ventilation. Because if you can imagine, all these aerosols that are suspended in the air around in this room would be greatly diminished if you had better ventilation and ways to control the ventilation in your space. So what are some ways to actually supplement ventilation in spaces? Well, I always say outside is better than inside. So that's the first step, because outside you basically have, almost infinite ventilation. You have the entire sky as your mixing room. Compared to inside, what I would suggest is increasing natural ventilation can be a really cheap and easy way to just increase ventilation to your space. But this can be especially challenging this time of the year because it's winter. And in which case I would suggest making sure that you're able to find ways in your room to increase ventilation somehow. So having HEPA filters or any type of filtration system in your HVAC will help with spreading in between rooms. But this is not actually going to decrease the aerosol of the people emitting in your room. Having a portable HEPA air cleaner in your room, however, will decrease that load of aerosols in your room. Another thing is ultraviolet germicidal irradiation, which definitely I'll go into some pros and cons of that as well. One important thing to note, which I know a lot of people have been wondering about for a very long time, what about plastic barriers? And really, plastic barriers only work if someone's going to sneeze in your face. Right? If we can think about going back to this mask, where aerosols can seep through the cracks in a mask, they can definitely find their way around plastic barriers. And what can actually happen is you might end up dividing the space in a way that creates these little eddies of higher aerosol concentrations compared to others. So really, plastic barriers do not protect against aerosols. It's really much more that direct, you're at the cashier, you're right in front of the cashier, breathing directly into their face or sneezing directly onto their face type of thing. So let's go in a little bit into ultraviolet germicidal irradiation. So they can be installed in a couple of different ways. They can be installed in the air handlers, but like I said, with HEPA filters in your air handlers, that's not actually going to decrease the concentration in your room, because that's not the volume of your room. It's more of between rooms that is going to impact. Another idea is putting one in the upper zone of your air. This should be facing upwards towards the ceiling, the light, and you should not have any potential of having this light go on to people, especially if it's the 254 nanometer wavelength. What's good about this is making sure that you have really nice airflow that kind of directs the air upwards and well mixes the room, so then you can have the virus basically being deactivated by these UV lights. Some important notes about UV lights. There's a couple different wavelengths. 254 nanometer wavelength light is the most common, and it is the most widely researched. So it can damage skin and eyes if there's a long exposure time, which is why really you should be installing these in the upper part of your room, where there's not going to be direct contact with people, and having shielding so people don't get exposed to this light. However, it has been shown to be really effective at decreasing any sort of viral load in a room. So one thing that's come out recently is 222 nanometer wavelength light. And what's great about this wavelength is that it does not cause damage to skin or eyes. However, it does produce ozone, and the amount of ozone that it produces is still being understood. I saw there's some questions about what about ozone generators. I do not recommend anything that says that it generates ozone as a plus is definitely not worth it, because ozone is it's great up in the atmosphere, keeping us safe from the sun, but it's very dangerous to your health, especially once the concentrations get too high. It can cause a lot of problems with your respiratory system, and a bunch of other things. So I would not advise having anything that says it may produce ozone. And also with the 222 nanometer wavelength light, there hasn't been that widespread on the market. So and there's a lot of, if you go digging on websites, you'll see some of them will say, oh, you have a strange smell. That's just producing ozone. It's okay, which is not okay at all from a human health perspective. So I would veer much more towards the 254 nanometer light compared to 222, and I would steer clear away from 185 nanometer light that specifically says it generates ozone, because it will just be harmful and produce a lot of ozone in enclosed spaces. One thing that I would recommend that's really easy to put into spaces are HEPA air cleaners. They've been shown in many different studies that they can decrease virus-containing aerosols in a room and decrease the transmission, because we're thinking about the room as a whole. One thing that you should be looking at in a HEPA air cleaner is called the clean air delivery rate, which is a combination of how how good is the filter in the air cleaner, as well as how much air actually passes through that filter. So then you can understand how much clean air are you supplying to this space. So for example, a CADR of 300 means that you have an air cleaner that supplies 300 feet of clean air per minute to your space. And what we're really thinking about in terms of air cleaners and ventilation is it's called ACH, or the air changes per hour. So how many times your room volume of air is being replaced in an hour. Ideally, the ACH should be above 3. In hospital settings, for example, it's closer to 12 air changes per hour, but that's also really expensive in terms of, the cost of actually heating up that air and making it a nice environment inside. So one thing that is pretty easy to do is figuring out how many air cleaners or how big of an air cleaner that you need based on how many air changes you want in an hour and supplementing your ventilation. Often these air cleaners will also say what effective room volume it supports. And it's really important to look at that because they're kind of taking into account how many air changes you're going to get based on what room size they think that it should fit into. Another thing I wanted to address was just this idea of expanding the space that restaurants take up. So I just wanted to say, a tent is very similar to an indoor space. It may be slightly more ventilated, but you're still going to end up basically having just an expanded indoor dining experience. And it's really important that people know that it's not the same as being outdoors. It's much more similar to being indoors. I've seen a lot of these tents, which I think are really cool because that's showing the pod that you're sticking with, you're really only exposed to that one pod. One thing that I've seen recently though, because it's winter, I've seen a lot of propane tanks being used to heat up these little tents, which can cause a lot of health problems for the people who are eating in these tents. You don't want to expose them to any sort of heating device that could end up decreasing the quality of the air that they're breathing while they're eating their food. I'm not sure how many of you guys have seen this recently, but CO2 monitors have become much more common thing to be searching on Google because they can be used to understand ventilation in a space. And this is because people, we breathe out CO2. So we're a major source of CO2 indoors. So we can understand approximately how ventilated a space is based on the level of CO2. If you wanted to measure CO2, you should make sure that you get it's called an NDIR CO2 sensor. And what this does is we can see, what is the approximate level of risk. So often they say you want it to be below 800 ppm of CO2, or at least below 1000 ppm of CO2 in a place to be better suited for people and decreasing the viral load risk. One thing that makes it really tricky though for people in the food industry especially is that there are many sources of CO2 that are not people. So for example, the oven and stove in my house are gas, and it increases the CO2 across the other side of the house by about 300 ppm just by turning the stove on. So this is something where you need to figure out what the background level is. But then also if you're using more stove, you may end up with more CO2 in a space. So this was a little bit trickier to use to understand how to increase ventilation or when you should do it, which is why I would suggest just making sure that you're able to have a comprehensive strategy for how to decrease aerosols overall. And to also note that many of the control methods that I showed earlier will not decrease CO2. So really, it's important overall to have a layered approach to controlling aerosols. So the main thing is increasing ventilation to a space, mechanical ventilation, if you're able to increase that, that's awesome. But also having open windows as much as possible is important as well. Using HEPA air cleaners is great. The portable ones make it very easy to add more to the space and decrease that viral loading as much as possible. making sure that everyone's wearing masks as much as possible, and keeping your social distance from other people, because those aerosols do disperse very rapidly with distance. So making sure that you're not right next to someone will definitely help. And that with that, here are my resources, and I'm happy to start answering some questions.
Fantastic. Tehya, Angie, thank you so much for your presentations. Every time we do one of these webinars, you know, if we were doing this in person, obviously we would all applaud and thank you for your time. And it doesn't have the same sort of payoff for that recognition, but on behalf of the audience members, and right now we're at like over 220 folks who are listening to the webinar and viewing it. Thank you so much for sharing this. We've had some questions come in through registration. We've had some questions roll in as through our presentation. So I've done a little bit of curation on these, and I want to start with something for both of you about droplet sizes and close contact and aerosolization all sort of wrapped up. So let me sort of capture this. Can you comment on particle size of respiratory secretions, and if and how they might decrease over time due to evaporation? And then could you also talk about the settling rate of particles and why smaller particles settle more slowly than larger particles, or if they do, and how that plays into our food settings, if at all? So sorry, that's like, there's a lot of things sort of wrapped up there. So hopefully you're able to follow my thread and parse that out.
Well, I'll just start because my comments on this will be quick. I'm going to defer to Tehya, who I think can comment with much more expertise about the physics of how these particles are working. But in terms of the virus, you know, there's some debate about where most of the virus is contained. And, you know, there's going to be issues of virus concentration. But also over time, as those larger droplets are evaporating, depending again on the circumstances, on the ambient environment, on the relative humidity, on the temperature, things like that, some of the viruses that are infectious will become non-infectious. And that really is going to depend on each individual situation. So in terms of talking about the physics of the particles themselves, the respiratory particles, that's not always going to be exactly aligned with how much infectious virus is actually in those particles and the infectious dose that people exposed to those particles are going to be exposed to. But I'll let Tehya take the rest of that question.
Yeah, so what we mean by droplets versus aerosols, there's a very fuzzy line between what falls within 2 meters and what stays suspended in the air very long, because the physics really depends on the situation that you're in. If you have completely still air, that's very different than having even small eddies in the air, but there's always going to be small eddies in the air. You know, there could be wind or a giant air conditioning unit that propels those droplets further. Let's see. There's a lot of complexity in the physics that makes it difficult to say what cutoff actually matters from like it being suspended in the air very long or not. So what you're actually producing standpoint from respiratory droplets, some of those droplets are, visible, like you can see them. So they're a few hundred microns in diameter. And some of those droplets are so small that you don't even know that they're there. What we've noticed is so when you project your voice, you're producing aerosol, a lot of that evaporation happens actually very quickly. And then they're just going to behave as particles of a certain size for like until they end up sticking to some sort of surface. But yeah, the physics is definitely quite challenging, but you do end up having larger particles, will fall at a higher speed it's based on their mass compared to the smaller particles. So.
Great. And as a follow-up to that, in the food sector, there's a couple of things that are happening that I really want to hit on here. There's a lot of air movement, and some of that can be forced air like away from product, and some of it can be air that's drawn. And so to give you an example of air that's being drawn, we would see that in restaurant kitchens pretty heavily. There's a a commercial hood that is looking to draw out odors and a whole variety of things that they would end up going through an HVAC system or directly out. Could you comment a little bit on how and this really it is kind of for definitely Tehya, I want you to talk about this, but Angie, I want you to, if you can, talk about how that might change the magic of the close contact 15 minutes 6 foot, where we've got this like just it's not a like static system of air movement. And so yeah, anyway, I would like to open that up.
Yeah, so I mean, this is a great example because we have seen situations in which airflow has actually contributed to transmission, specifically in food settings in restaurants. And in particular, last spring they published, or a preprint was published about a restaurant in Guangzhou, China, in which an air conditioning unit basically blew air and presumably aerosols and droplets to tables that were essentially downwind from the person who was infected and dining. And that exposed all the people within that line of airflow to infection. And when they did the epi-analysis of it, they showed that only people who were basically sitting in that current of winds from the air conditioner were infected. So a lot of this though is multivariable, right? Like you have to have all the conditions be just right. You have to have that airflow pointing to other people. You have to have a person sitting upwind of those other people who is shedding enough virus that those people are getting exposed to a dose of it across that distance. You have to have, if you don't have somebody who's shedding enough virus to say that a very brief interaction is going to result in an infection, you still have to have them for a long enough period of time exposed to those other people in that current of air to give them cumulatively enough virus that they're going to get infected. So it seems deceptively simple, but it's actually really, really complicated and context dependent. And I think that's why I think a lot of people have focused on the CDC's definition of close contact, this 6 feet, 15 minutes rule. And I think that it's dangerous for people to think that that's a hard and fast rule, because it's very dependent, again, on all these other variables. You can certainly be exposed to somebody for a much lower period of time for less than 15 minutes, and you can transmit the virus. You can also have people who are more than 6 feet apart contract the virus as well. A lot of it just really depends on the circumstances that are leading to exposure. So I think that, the definition of close contact is not so, I think it's become what people are thinking of as guidance. But the real reason for that is for contact tracing, because if you were trying to do contact tracing and then you traced every single contact that you even had very incidental, brief contact with, that's going to become very quickly an impossible task to manage. So contact tracers use these types of definitions so that they can manage that most effectively to look at the most high risk contacts, but that by no means, establishes some sort of magical parameter below which you can't get infected. You can certainly get infected through a brief interaction with somebody if the conditions are right.
Thanks for that. I know we're getting close to our time here, and I wanted to, especially Tehya, for you to comment if there are a low cost but still effective measures that some food producers who may not actually be able to impact their own HVAC systems, but can employ in a food setting to reduce risk. And things like air sanitizers, is that something that is reasonable? And we've certainly seen a lot of marketing in that area, and just wanted to open that up of like what are practical things, and what types of things should we be wary of?
Yeah, so I would just make sure that that you are looking at, reputable sources. Don't trust anything that says that it seeks out viruses and destroys them. Like, you're not just, what if you destroy something, what are you breaking it up into smaller particles, in which case, like, you have even more particles to worry about? Like, I don't know what that means. Main thing, if you air cleaners are relatively cheap, relatively cost effective. As long as you size it appropriately to your space, you can dramatically decrease the risk to everyone in the room, just of that aerosol buildup. HEPA air cleaners have been shown to be very effective at decreasing viral load in a space without any adverse effects, such as like creating ozone or anything like that. If you are interested in UV germicidal irradiation, that's also an option. It is, however, more expensive, and the understanding of how to install it is more complicated. And you might be also sent down a few more rabbit holes of people trying to sell you things that are not actually feasible or produce ozone unknowingly. And then you end up with employees who are coughing all the time because of the ozone. So I would say biggest things, when it's warm outside, always, try to do things that are outside, increasing natural ventilation as much as possible, and HEPA air cleaners. And then, of course, making sure that people are distanced apart from each other, that there's not really that directional air that we were just talking about in between tables, and knowing that really those plastic dividers aren't actually effective at decreasing aerosol risk.
And if I can just jump in really quickly and add something here, and that is that the problem with UV germicidal irradiation is that it's not regulated at all. And so there are a lot of different products out there of varying quality. The other thing people should keep in mind about that is that the way that germicidal irradiation works is by creating covalent bonds and dimers between different nucleotide bases in the viral genome. And that has to occur frequently enough within a given viral genome to actually make that genome non-functional. So that is really dependent on exposure time and distance. And some of these products, for example, we use germicidal irradiation to sterilize our hoods. We also still wipe them down with surface disinfectants when we're working with viruses, because over time, the irradiation loses its potency. And it also is impacted by distance from the UV radiation source. So, people can look into that, certainly, but as Tehya pointed out, they're expensive to install, and there are a number of other options that are better regulated and that we know will work and are also cheaper to implement.
Wonderful. I just want to thank you both for your insights today, for your time, for our audience to join us. There were probably 30 other questions we couldn't get to, but for us, as the Food CoVNet project, we'll answer these questions as a companion piece with this webinar. We'll have answers to these posted on our website. So we'll follow up with all participants who registered, because we have your emails, and we can let you know when those questions are answered. I'll share my screen really quickly again here. Just to thank both of you again for your time. And I really wanted to just highlight that for more information on all of this, you can go to foodcov.net. We have future topics coming up for other webinars in this area on environmental monitoring, supply chains, and investigating transmission routes, and then also COVID-19 in the food system and trade impacts. So please, thank you again for joining us. But if you do have additional questions or topic areas you'd like us to cover, please don't hesitate to reach out. And again, Tehya, Angie, I can't thank you enough for you engaging in this dialogue and bringing your expertise to our group. And again, we'll follow up with you to help us answer some of these questions that our audience provided. But again, thanks so much.
Thank you, Ben. This was really, really great, and I learned a lot from Tehya.
Yeah, me too. It was fantastic. All right. Thanks everyone. We'll see you next time.
Take care.
Thanks.